Last night we were on YouTube in my house because my children wanted to listen to David Bowie, which I think is awesome. And then I was like, hey, I have a YouTube channel. And they looked at it and they were like, can we watch something else? So clearly not engaging for the 5 and under set. I think they expected more of a David Bowie kind of flair. I know. Well, my husband was like, what, we can't see you? I was like, no, you can't see me. You can only listen to my bad jokes. Okay. Let's move on. So we talked the other day about just the basic concepts with cell cycle and the checkpoints and how going through the different phases of the cell cycle is regulated by a bunch of kinases called CDKs, cyclin-dependent kinases, and they're controlled by cyclins. Makes sense because they're cyclin-dependent kinases. So they're controlled by the availability of cyclins, and it's this this fluctuation of cyclins that regulates CDK activity. So, what did I just say? Cell cycle regulation depends. This is where we ended the other day. Cell cycle regulation depends on the activity of CDKs, cyclin-dependent kinases. So here, the red one's your CDK, and here's your cyclin. And when the cyclin binds to the CDK, CDK is active. So you can make the assumption that if there's no cyclin, the CDK is off, right? And so that's how cell cycle is controlled. When you need a CDK turned on, transcription, translation, increase the amount of a certain cyclin, that cyclin's there, it can activate the kinase. Kinase can go on and phosphorylate things that are specific to that part of the cell cycle. And then when that cell cycle is over, that part of the cell cycle is over, then you degrade your cyclin by ubiquitination and proteasomal degradation, and then you move on to the next phase of the cycle. That's a very simple way of saying it. Okay, so right here I have it saying these kinases, the CDKs, control the activity of different proteins involved in controlling the major events of the cell cycle. What are the major events of the cell cycle? Someone give me some major events. Cytokinesis. Cytokinesis, that's a major event, I'd say. Yep. What else? Organelle duplication. Probably, but I don't know the exact mechanism off the top of my head. But you're probably right. There is something going on there. Yep. Formation of the mitotic spindle. So centrosome duplication. Formation of the mitotic spindle. Okay, what else? Big ones. There's some huge ones. Yes. Right? Chromosome segregation. That's a huge one. What else? What else happens during the cell cycle? Major events. Someone else besides the 2 or 3 people. Yeah. Right. So DNA duplication, DNA replication. Right? And we're going to learn about that today. So all these things are controlled by phosphorylation by a CDK. Okay? If you don't have a CDK that's going to phosphorylate certain things in chromosome duplication, you won't get initiation of replication. You won't move through S phase. Okay. For chromosome segregation, you need certain things to be activated to degrade certain parts that keep the chromosomes stuck together because you can't separate them when they're stuck together. Any other major events of the cell cycle people can think of? I think you guys got the big ones. Big ones. Okay. Good. So this is, you saw this the other day, right? So the cell, how does the cell use the regulation of CDKs by cyclins to control the cell cycle? Well, the CDKs, the cyclins are going to increase in abundance and decrease in abundance, and that's going to control the activity of the respective CDK. So there's a CDK for G1 S phase. There's a CDK for S phase. Now this is very, very simple. There's actually a ton of CDKs, but we're putting them in very basic categories. So you can think, well, SCDK is going to be involved in S phase. M-CDK is going to be involved in mitosis, because if I start giving you things like cyclin D1 and all these crazy alphabet soup names, then you're going to start crying and kill me. So we're not going to do that to you, at least not until you go to grad school to study cell cycle. People who work on cell cycle were not, bless you, very creative in naming things. Everything seems to be like a CDC something or a CDK something or a P21, 22, 23, 24, 25. So, anyways, so cyclins, they rise and they fall throughout the cell cycle, and they activate the CDKs, and that directs them to the correct target proteins, right? So the CDK is specific to a certain target protein, and when it's active, it'll go and phosphorylate that target protein, and that will affect its activity. Okay? Basic concepts here. I think we're good so far. Any questions on these basics? I've been grading the exams, and we're getting close to the end, happy to say. And what I've learned from grading them, people have been doing fantastic, which is great. But what I've learned is there are some people who never got the concept of dynamic instability or GTP cap. And that bothers me only because I stand here and I'm like, does everyone understand? Does everyone understand? And then like, well, if you don't understand, I know everyone's shy, but I can guarantee for every time you raise your hand, 20 people are going to say, thank God she asked that question. Or he asked that question. Okay, so everyone gets this concept of the fluctuation of cyclins and regulating CDK activity, right? Okay, and that the kinases are phosphorylating proteins involved in progressing through different major events of the cell cycle. You got it? So if I give you a quiz right now, you can tell me about that? Okay, we'll see. Okay, so there are many levels of regulation of CDKs. So it's not just about binding to cyclins. That's the basic idea. But there are many levels. And this is because you need to assure tight regulation of the cell cycle, right? If there's one way to regulate entry into mitosis and something goes wrong with that one way, is that good for the cell? No, right? You want to have many levels of control over something as important as the cell cycle. If the cell cycle becomes dysregulated, what happens? Is all well? We're happy. We get to go to the state fair. What happens if the cell cycle becomes dysregulated? Cancer. Right. Cancer. That's the answer to everything. That or ubiquitin. Right? So different mechanisms, that is so loud. Different mechanisms for regulation of the cell cycle include, like I just said, the availability of the cyclin, right? Okay, if there's no cyclin, you can't activate your CDK, you can't phosphorylate whatever target proteins to enter whatever phase of the cell cycle that you need to enter. Availability of the target protein, right? If the thing that needs to be phosphorylated isn't there, then you can't phosphorylate it, and then you're not going to turn whatever on, right? So proteins, for example, proteins required for mitosis will only become available in G2, right? So you have to think about this. There's phosphorylation going on to activate things, and then there's this fluctuation of protein levels that helps to regulate the activity of things, right? So as your cyclins go up, your CDKs become active or inactive. But then there's also the increased expression of things, right? Because if you're going to degrade your cyclins, you need to increase their expression again in order for them to be available when you're going to need them. Another mechanism of regulation, which we'll talk about, is inhibitory phosphorylation. So you remember, phosphorylation is not always about turning on. It could be about turning something off. And so there's inhibitory phosphorylation of CDKs. So this is phosphorylation within the active site. If the active site of a kinase is phosphorylated, it can't phosphorylate, right? It's like throwing something into the gears, right? Activating phosphorylation, so that's a kind of phosphorylation that's activating. And then there are these CDK inhibitor proteins, CKIs. And these are proteins that bind and inhibit the cyclin CDK complex. So these are a lot of different mechanisms of regulation, right? But they all make a lot of sense, right? If you don't have your cyclin, you can't activate your CDK. If you don't have the thing that needs to be phosphorylated, you can't phosphorylate it and it can't go do its job. If you phosphorylate a CDK in its active site, it can't go phosphorylate things. Yes? Did they go out?
Yes.
The bathroom used a if they need to be phosphorylated to be active, that's another way to regulate. And then there are these proteins that bind and kind of sequester active CDK cyclin complexes. Okay? Here's a nice figure that shows it all. Okay, so what was she just talking about? I don't know. Who knows what I was just talking about? Is everyone still thinking about microtubules? Okay, where's the treadmilling? No treadmilling. Okay, good. So CDK activation, right? Cyclin binding induces a conformational change in the CDK, and that allows for an activation, an activating phosphorylation to happen, right? So you've got your CDK, it's inactive. This red thing is just kind of an illustration of part of the protein structure that changes when cyclin binds. So cyclin binds, and so it goes from this conformation to this conformation. This little loop sticks out. And that allows for CAK, CDK activating kinase. What's CAK going to do? Phosphorylate. Is that activating? Thank you. I just want to make sure that people remember CAK has the word activating in it, so it's going to be activating. Phosphorylates the CDK, and then it's a fully active cyclin CDK complex. So you can imagine that if you don't have your cyclin, it's not active. Also, if you don't have your CAK, it's not active. So there's two levels of regulation of that CDK right there. Okay. Why does this matter? Why am I going on and on about regulation? It's just the beginning. It's going to go on and on for days. Days. And you can't drop the class anymore. You're stuck with me. Why am I like, wow, you need to regulate this cyclin CDK complex. This CDK needs to be regulated. Why is this so important? Why do I care? Why do you care? Other than that you want to pass so you can graduate. Right? So if you don't have tight regulation, important functions of the cell are messed up. Right? So things aren't activated properly or inactivated properly. So you'll go into S phase when you shouldn't. You'll go into mitosis when you shouldn't. Or you won't go into S phase when you need to. All these fun things. You'll segregate your chromosomes before you're ready. Things that are bad. Okay. Negative regulation of cyclin CDK by inhibitory phosphorylation. So I just showed you activating phosphorylation by this CAK, cyclin activating, what is it? CDK activating kinase. And then there's negative phosphorylation, and this is right in the active site. So here's your CDK with your activating phosphate. It's bound to a cyclin, right? So this thing is ready to go. But WEE1 kinase, which is an inhibitory kinase, can come and phosphorylate right here in its active site, and that prevents this cyclin CDK complex from phosphorylating anything. And let's say you want to turn it on, you can activate a phosphatase, CDC25, and it'll remove that phosphate group, and then it will be active again. So why would you want to have this level of control versus just controlling levels of cyclins, right? I said, well, cyclins go up, they bind, they activate, and the CDK is ready to go. Why would you also want this inhibitory phosphorylation, dephosphorylation? What's the point of it? Other than it just being another level of regulation. Yep. Yeah, so that's one thing. You don't need to make more cyclins. Like, you don't need to make new. So remember we talked about in the past, phosphorylation versus increased gene expression. So if you need to make a new protein to associate with your CDK, then you need to, transcription, translation, whatever modifications a protein needs to be active, proper folding, right? That takes a lot longer than adding a phosphate group or taking off a phosphate group. So here you can have a gradual buildup of cyclins, and then that CDK complex can be kept inactive until you activate your phosphatase, right? So then you have a large pool of cyclin CDK complex ready to go as soon as it's needed. Questions? I find this stuff riveting. Who finds it riveting? Yay! I love it when people actually give me a thumbs up. Okay, good. Okay, so I talked about negative regulation by CKIs. These are cyclin kinase inhibitors. Cyclin kinase inhibitors. And what they do, right? Remember, so you've got your CDK bound to your cyclin. It's been phosphorylated. It's active. This thing is ready to go on and phosphorylate things. Well, it can be inactivated by a CKI, in this case P27, and it will bind to the whole complex and distort the active site of the kinase. So it prevents the kinase from phosphorylating things. And so this is another way. So if you have, we just had keeping it inactive by phosphorylating in the active site. So here you can keep it inactive by binding to the whole complex and preventing it from interacting, distort the active site and preventing it from interacting with its substrates. So this is another quick way to regulate things, because as long as P27 is there, it's going to bind and prevent, and then at certain times you'll degrade your CKIs when you want to activate things. Yep.
So which state is more common, inactive and phosphorylated, or inactive, unphosphorylated?
Which state is more common? Inactive and phosphorylated, or active and inactive unphosphorylated? Okay, wait. Say that again.
I was wondering which state is more common in the cell.
Oh, you mean this one or the other one?
Yes.
I think that it's not about one's more common than the other. It's just that they're all happening. Yeah. So, I mean, I guess we could, yeah. No, it's just that there are just many different levels, and depending on the process, because there's so many CDKs, right, and so many cyclins, and so they're going to be regulated by all these different mechanisms. Okay? Yeah. I'd say they're both common. Okay, so the whole point of the cell cycle and regulation of the cell cycle is that it's controlled by transcriptional regulation and cyclical proteolysis, right? Transcriptional regulation, what does that mean? What's transcriptional regulation? Yep. Right, so regulating gene expression, how much of a certain gene will be expressed and how much protein will be made, right? And cyclical proteolysis, what does that mean? Nothing?
Proteins?
Destruction of proteins, what does cyclical mean? In a cyclical fashion, right? That was that up and down kind of thing that I was showing you, right? So transcriptional regulation, so we're increasing gene expression. What kind of genes are we increasing the expression of? I keep talking about degradation. Oh my God, gene expression. What kind of genes are we expressing here? What kind of protein rises? Cyclins. So that's one example of, remember, it's not just about degradation. Proteins come from somewhere. So you have to think of the whole story, right? So if you're going to degrade at the end of each phase of the cycle, if you're going to degrade a whole bunch of cyclin, you're going to have to increase the expression of those genes when you're going into the next one, the next phase of the cycle, right? Or the next time around, I should say. Right. So what types of proteins? So this is how cell cycle is regulated. If you have to remember one thing, it's that rise and fall of proteins, and that it's controlled by increased gene expression when you need a protein, and increased degradation when you need to turn something off. That's one thing that you need to. If there's anything you take away, that's it. So what kinds of proteins do you degrade? So we just talked about how we degrade what? Cyclins, right? Cyclins. We degrade cyclins. What else might we degrade if we need to turn something off? Or let's say we want to turn something on. What might we degrade? CKIs, right? CKIs are keeping a cyclin CDK complex inactive. We might degrade CKIs. What else might we degrade if we want to keep something off? So like if you think about it, I just told you all these ways of regulating. One was inhibitory phosphorylation, right? And so if you want to keep it phosphorylated, you're going to turn off gene expression for the phosphatase and degrade all the phosphatase around, right? So you always need to think about things. When I ask these kind of like very open-ended questions, think to yourself, okay, what are the levels of regulation? What are the proteins involved? And how would increased gene expression or degradation affect that? What types of genes might be upregulated? We just talked about one, cyclins, or CDKs, right? Might you need to upregulate a CDK in advance of needing it, right? Okay, big picture questions. Not much excitement here. Okay, we're gonna keep going. Okay, so I'm talking about regulated degradation of proteins, right? It's not just cyclins that are being degraded, right? Like we just spoke about. Here's an example. Well, here's an example of M-cyclin being degraded. But I want you to understand that there are different ubiquitin ligases that I'll introduce right now, and they degrade many things, not just cyclins, okay? So the first one we're going to talk about is APC, anaphase promoting complex. So you might think it might have something to do with promoting anaphase at some point. It's not only going to do that, but anyways, we'll talk about that later. So it induces degradation of securin, and that's anaphase promoting complex, right? Securin keeps sister chromatids together, and so you degrade that, and then anaphase can happen. So that's one way you can remember one thing. It degrades germinin, which we'll talk about in a minute, and it also degrades S and M cyclins. And the way it works is you need to activate your ubiquitin ligase. So you have your inactive APC, and then this activating subunit, CDC20, will come along, bind to it, and it becomes active, right? Because you don't want APC to be active all the time, right? What would happen if APC was active all the time? Would you ever go through M phase or S phase? No, because you would always be degrading S and M cyclins. So you activate your ubiquitin ligase, and then it does E1, E2, transfer ubiquitin, polyubiquitin chain to M cyclin here, and you get degradation of the M cyclin, and then what happens to that CDK? What happens to the CDK when you degrade your M cyclin? Inactive. It's inactive, right? And that's how you turn off your MCDK. You degrade your M cyclin. And as you'll see shortly, there are a whole lot of things that are degraded, and it'll all start to make sense shortly. So another ubiquitin ligase that is involved in cell cycle is the SCF ubiquitin ligase. And it's called SCF because it's got three components called skip, colon, and F-box. So there's your colon and your skip and your F-box protein. It's SCF. It doesn't matter. It's important to me because I work on colon, but I won't take offense if you guys can't tell me all the subunits. So that ubiquitin ligase is involved in degradation of CKIs. So you have your CKI, and it could be phosphorylated, which targets it for degradation. Because the F-box protein is the part that binds to the substrate, and it only recognizes phosphorylated substrates. So that's a way of controlling the activity of the SCF ubiquitin ligase. Remember, we control the activity of APC through having an activating subunit, and here we control activity of SCF by making the substrates more appealing or less appealing by phosphorylation, right? So here this substrate is going to be very appealing because it's been phosphorylated by a kinase, and then it'll be ubiquitinated and degraded by the proteasome. And so what does this do to the activity of the cyclin CDK complex that it was binding to? Yes? It activates it, right? CKIs are inhibitors. The I stands for inhibitor. When we degrade our inhibitor, it that whatever was being inhibited is no longer inhibited, right? Okay, so there are many substrates of SCF. Those are your two ubiquitin ligases involved in cell cycle. There are many substrates of these ubiquitin ligases, and the substrate degradation depends on what ubiquitin ligase is activated when. And so I like this. It's very complicated because it shows you all the substrates, but I like it because it shows you all the substrates in terms of when the ubiquitin ligases are activated and what's being degraded. Because here's your cell cycle, right? And the APC is red and the SCF is blue. And you can see that APC is only active during this phase of the cell cycle. And that's on purpose because there are times that you need to degrade certain things and there are times you need to let things accumulate again. And that's really important. So that's what causes that cyclical proteolysis that regulates the cell cycle. Okay, so it all goes back to ubiquitin ligase activity. You guys are all going to leave here going, ubiquitin does way more than just degrade misfolded proteins, right? Remember thinking that? How misled everyone was. Anyways, so there's the SCF, which is involved in degradation of a whole bunch of things, and the APC that's involved in degradation of a whole bunch of things. And some things you could see in here are securin, cyclin A, B. See, the cyclin names, there's a lot of them. CDC20, geminin, SCIP2, which is actually, SCIP2 is actually part of the SCF ubiquitin ligase. Cyclin D, MYC, June, CDC25 is degraded by SCF. CDC25 that was involved in, wasn't that the phosphatase that removed the inhibitory phosphate? We1. So a lot of these things that we just learned about, all these different proteins I told you about that regulate cyclin CDK activity, they're all regulated, their levels are all regulated by these ubiquitin ligases in a cell cycle dependent fashion. Okay? Any questions? I know I'm going on and on and on about degradation. I just can't help myself when I talk about ubiquitin. Yes?
They regulate the levels of the
Proteins. See all these proteins? All of those. So, but the key is that they're regulating levels of proteins in a cell cycle dependent fashion. So as you go into a certain cycle, a certain phase of the cell cycle, if a ubiquitin ligase is active, it might be degrading things that might be inhibitory to entering that cell cycle. And as you leave it, it might be degrading things that were involved in keeping that part of the cell cycle on. Okay? Yep. Why is it so active during G1? That has to do with its targets. Because, so there are a few things. So we'll talk in a minute about geminin, but geminin is involved in origin licensing in preparation for S phase. So you want geminin is an inhibitor, actually, of the beginning of DNA replication. So you actually want it to be off after you've just regular. You actually want it to be on, so geminins turned off. And then securin, really important for keeping chromosomes together. So you want to degrade that going into M phase. I mean, really, you could say that. So a lot of this is things that need to be turned off as you exit S phase, and things that need to be turned off as you enter M phase. And then you would say that there's an equal amount of things being degraded on the other side in G2 and S, right? Did that answer your question? More questions, thoughts, opinions? Nothing. Okay. So why so many levels of regulation? Why not just control cyclin synthesis and degradation? Well, we just talked about that at length, right? Hopefully you all can talk about this, right? Hopefully you all can think about this right now. And you're like, well, she just told us this, right? Why so many levels of regulation? Because it's important. It's important. You don't want your cell cycle to be dysregulated or you'll end up with cancer. Well, in the event that apoptosis doesn't work, cancer. But usually apoptosis works unless you're really headed for cancer. Why not just control cyclin synthesis and degradation, right? Why not just do that? Why all these other levels of regulation, dysphosphorylation, and these proteins that bind to things and inhibit them? Why?
You may not want to degrade your cycle. You may want to keep it there for future reference.
For future reference. You might not want to degrade your cyclin. You might want to keep it there for future reference. So if a cyclin's involved in activating a certain phase of the cell cycle, why would you want to keep it around? Well, maybe you're not.
Yeah, save energy.
Well, usually once you are leaving at that phase of the cell cycle, you don't keep things around. But don't forget, when you start to accumulate your cyclins, it's not just bam, so things start to accumulate prior to activation of that phase of the cell cycle. Did I see your hand raised?
Yeah, I was just going to say it might be good to keep them around so that they're already there and ready to start entering.
Right. So as G1, so as you're going into S phase, let's say, you're going to have an accumulation of your cyclins before then, but you'll want to keep them off until you're ready for activating, for triggering S phase. Okay, so these are all the different cell cycle regulatory proteins. Not all of them, but the major ones. We just talked about some. They're in the book, but their main classes are protein kinases and phosphatases. Yes?
So the answer to the second question, would that be because it'd be less efficient to keep synthesizing and creating them and do more energy? So it's more efficient and less energy to keep them around and just regulate it?
Yep. It's more efficient to keep something around and regulate its activity rather than constantly degrading and transcribing and translating and making more protein. Yep. And with that, the fact that the buildup of a lot of these proteins happens over time before it's actually needed. So you might keep things inactive until they need to be activated. Yep. Yep. Well, so what you said is that you are under the impression that cyclins are degraded at the end of the cell cycle. So would this be applicable just for cells that are replicating often? No, it's not only about cyclins. Remember, it's not only cyclin that's degraded. Well, let me go back to that. It's not only about degradation at the end of a cycle. So you're right. As you leave a cycle, you get degradation. That's absolutely right. But it's not like it's black or white. You have this slow build, and then you have them stay around, and then they go down. And this slow build, that slow build might start like half a phase before you get to where you need that cyclin or that CDK. So, yeah. usually you degrade your cyclins when you're done. Yep, and it seems like a huge waste of energy, but it also seems like if you're going to waste energy on anything in the cell, waste it on maintaining your cell cycle, right? Because it's a lot worse to be apoptosing yourself all the time, right? Apoptosing? Killing yourself? Okay. Good questions, guys. Okay, so the different categories of proteins involved in cell cycle regulation are these kinases and phosphatases that modify CDKs, CKIs, the inhibitor proteins, and ubiquitin ligases and their activators. So if you're looking at categorizing all this information I gave you, here's a way to start categorizing in terms of regulation, right? So things that phosphorylate, things that remove phosphates, things that are CKIs, and ubiquitin ligases and mechanisms for activation. Okay, so here's the overview. So here's your whole cell cycle, right? G1S, G2M. You can have external effects, which will affect the activation and transcriptional regulation of things. So if you have a favorable cellular environment, then you'll activate your G1 CDK, and you'll get G1S cyclin synthesis, and you'll start G1, right? And so that's kind of the beginning of your cell cycle, right? And if you have any of these bad things like DNA damage, unreplicated DNA, right? These are these checkpoints we talked about. They're actually going to inhibit a lot of these things, right? So don't forget also inhibition is also about turning things off when you need to stop and fix things, right? So then you'll get G1S CDK. Then you'll have S CDK. So if these bad things don't happen and you want to proceed through your cell cycle, then you'll get S CDK activation, right? And then S cyclin synthesis starts over here in G1, and there's this slow build, and then it binds CDK and activates and turns on S phase. You'll get your DNA replication, and then you'll get your M cyclin. Where is that? That's not on here. So your M cyclin is going to start to build up in the end of G2 as you start to enter M phase. That'll activate your MCDK. Again, unreplicated DNA damage, not going to allow MCDK to become active because you don't want to go through mitosis if your DNA is not replicated or if it's damaged. And then MCDK is involved in activation of APC, the ubiquitin ligase, and that's involved in degradation of geminin, which we'll talk about shortly, securin. Securin's the one that holds the chromosomes together, and S and M cyclins. And that is important for exiting mitosis. If you don't have activation of APC, you don't exit mitosis. Okay, so let's talk about S phase, right? This used to be a blank slide, and I was like, I need a picture of S phase. Do you think that was a good one? I was very frustrated with this picture of S phase because it's not showing any proteins on it, but that's okay. So, S phase is regulated by two things, CDKs, S CDKs, and S cyclins, and APC. Okay, so this is the overview of what's going on in terms of your DNA during the cell cycle, right? So in G1, you get this pre-replicative complexes that form at your origins. And then when SCDK is turned on, that results in the actual replication start happening. And so then you get your replication, and then MCDK activation gets turned on, and then you get your chromosome segregation, and then APC activation is activated, and CDK is in. I don't know what that means. And the MCDK is inactivated, and then it starts all over again. Okay, so I'm gonna actually focus on this phase of the initiation of S phase. So first you need to think about initiation of DNA replication. It all depends on the assembly of your origin of replication. If that doesn't happen, then your helicases aren't loaded, then you don't open your DNA, and then you don't open your DNA, then you can't replicate it. Does everyone agree with me on that one? Great. So here's your origin, and these proteins, CDC6 and CDT1 come, they bind, and then a helicase comes, and this whole point of this whole origin of replication complex is to help to load the helicase, and that's the pre-RC. So now you have your origin loaded with a helicase. SCDK then comes along, it's activated in S phase, and that results in disassembly of this pre-RC, because once your helicase is loaded, all those proteins need to go away because they're going to interfere with DNA polymerase coming along and doing what needs to be done, replicating DNA. And so then you get induction of DNA replication. So this ensures that DNA replication only happens once per cell cycle, okay? Because what's happening is you're having control of the ORC and the pre-RC complex. If that can't assemble, you can't replicate your DNA. And so what do I mean by that? Well, the points to kind of focus on are that when you have SCDK activated, then you get disassembly of this pre-RC because you get phosphorylation and degradation of CDC6, and then the CDT1 gets degraded also. Not degraded, inhibited also. And we'll go into more detail right now. Okay, so the ORC and the pre-RC, it has to assemble to load your helicase at the origin. And for that to happen, this protein called CDT1 needs to be available, okay? So, oh wait, you guys saw this before. A bunch of you took me with 4-9. I go over this in 4-9. You guys are all gonna have an easy time right now. So CDC6 and CDT1 assemble on the origin, and then your helicase comes, and that's your pre-RC. So now let's talk about APC. Okay, APC during this, that's the ubiquitin ligase. This is active at this time, and it's actually degrading geminin, and that's what allows CDT1 to be active. Because CDT1 is normally bound by geminin because it's an inhibitor. Geminin's an inhibitor of CDT1, and so this is how APC activity regulates formation of these the pre-RC and initiation of S phase. At the end of G1, stop it. What happened? I zoomed. Are we good? Cancel. Cancel. I've never done that before. How about select? No. How about, where's the clear button? Cancel? What's going to happen there? Ah! I broke it. I guess, well, you guys have me for 5 more minutes no matter what. So are we going to have to do the whole thing? It's super huge? No, unzoom. Unzoom. Oh. Yeah. Woo! Okay. Okay. So APC is inactivated as you... Wait a minute. APC is active here, so geminin's not around. So geminin cannot inhibit CDT1, so you can have this pre-RC complex formation. At the end of G1, you have APC inactivated, which allows geminin to accumulate again, and it binds to CDT1, so that inhibits pre-RC formation. Okay, so Then S phase comes along, right? SCDK comes along during S phase, right? So it targets CDC6 for degradation, and it activates APC. So CDT1 is inhibited by geminin, and then the pre-initiation complex comes along and binds, and you can get DNA unwinding and initiation of replication. And and you have disassembly of this pre-RC. So this is why you don't get initiation of S phase again. So at the end of mitosis, actually, APC becomes active again. And this allows for degradation of geminin. And so then you can start to reactivate it. Allows for degradation of geminin. And so you have inhibition. So you have activation of CDT1. So CDT1 can go and start to form a new pre-RC. And you'll prepare for entering S phase again. Any questions about that? So it all has to do with APC activation and assembly of your pre-RC complex, and allowing geminin to be degraded when CDT1 needs to be available for pre-RC formation. Okay, so here are a few practice problems for you guys to think about for what we started with. Let's just go through this together for the last few minutes. Okay, so how are cyclins regulating the cell cycle? You want to take a minute to talk to your friends? 1 minute? You should all know it.
Well, they find the CDK well, they call them to call out
Okay, guys. How do cyclins regulate the cell cycle? Accumulate, bind CDKs, activate. How do cyclins? So that's how they turn on a phase of the cell cycle. How do they turn off a phase of the cell cycle? They're degraded. Yep. Or they could also be phosphorylated. They're not phosphorylated to CDKs. That's phosphorylated. Okay, so you suspect. Okay, let's go through this one, and then you guys can go away. You suspect that a defect in protein X has a negative effect on cell cycle progression. Okay, protein X, something's wrong with it. It has a negative effect on cell cycle progression. How would you use the tools explained in class, remember Monday, to evaluate whether there's a defect in cell cycle and where that defect might be? Yes?
Don't you use like the temperature sensitive mutants? Like you can turn it on at certain points to see if it's reactive.
Okay, so there's a few things. So temperature sensitive mutants. So let's say that you have a temperature sensitive mutant of protein X. Okay, so now you can increase the temperature, see your defect, and how would you be able to evaluate where that defect is? Yep.
If one protein X is active and upregulated, you would see cell cycle arrest in some part of the
Active or inactive?
Well, it says it has a negative effect on the progression, so wouldn't it kind of come to a halt?
The defect has a negative effect.
Sort of
So cell cycle arrest. Okay, so you'd look for cell cycle arrest. What tools that I talked about on Monday would you use to evaluate? Yes.
Is it the I can't remember the name of
The PI?
the yeah,
PI staining. So what is PI staining? What does it look at? What is PI stain? Propydium iodide. What color is it? Orange. So you remember orange. What was orange when you looked at the cell? DNA. Right. So how does the intensity of orange staining change through the cell cycle? When is it lower and when is it higher? Higher in S phase, right? Because when you double your DNA, you're going to double your intensity of your staining. Okay, so you can use PI staining and your temperature-sensitive mutant to see what phase of the cell cycle you're in, right? Because let's say it's a defect that prevents DNA replication, then you won't see any intensity, right? It would all be 1x. Or you can use flow cytometry. Well, that's flow cytometry for that. Or you can use BRDU, which is looking just at DNA replication. Okay, you can leave. We'll finish this next time.